I'm Creighton Miller, and I'm with the Horticulture Department at Texas A&M University, which is a part of Texas AgriLife Research. What we're reporting on at this meeting is a breeding methodology which we call Fast Track. We've searched more than 600 different varieties and selections to try and find zebra chip resistance or tolerance, and we're still searching, but we're finding some that are certainly more tolerant to the ZC than others. We're pretty certain that when we find what we really are looking for, that it may not be a variety or a selection that is totally appropriate for chipping, and that there may be some further breeding. So what we've proposed is a fast-track breeding technique to help facilitate that. In breeding new potato varieties, we generally expect that it's going to take somewhere between 10 and 14 years. And we've carefully looked at all the steps in a breeding program and tried to examine where we could shortcut that. One of the things that we have to do when we make a selection in a breeding program is clean it up of any viruses or diseases that the selection may have. And that generally occurs 2 or 3 years after the selection is made. And so what we've proposed to do and what we demonstrated could be successful is to shortcut that. And so we picked out one cross that we had, which was between a regular average chipping variety and one that we had identified as having some ZC tolerance, namely NY138. So we crossed those two, and we planted the children from those crosses out in the field. And from those children, from those two parents, we selected some 32 individual tubers or children from that cross. And we brought those in, we put them in cold storage to break dormancy and get them to sprout. And then took them out of storage after about 65 days and allowed the tubers to sprout. We took small cuttings off the sprouts on the tubers and introduced those into tissue culture. So right there at the very beginning of the whole process, we already have each of those 32 children in tissue culture. As it turns out, we tested them for all the viruses, and all the children were clean of PV potato virus Y and the other viruses. Then we took each of the 32 tubers, cut them into 4 seed pieces, a 4 cut on the tuber. We planted 2 of those in cages down in Weslaco, and then we planted 2 of them in a commercial field nearby in Edinburgh. We grew those plants up in the cages and in the field, and then went in and harvested them, and then chipped them and evaluated them for normal chip characteristics in the tuber and for zebra chip. From those 32, we found 2 that we advanced from that point. And we're also, when we put them in tissue culture, we are also growing mini tubers from those tissue culture samples. So the bottom line is, within 1 year from the time they were selected in the field and evaluated, we had identified 2 out of that family of the children that passed the test for both zebra chip and quality characteristics. This probably cuts off 4 to 6 years in the testing process, and we have not done heritability studies yet on ZC resistance, but we're assuming that there is a degree of heritability for this trait. We're fairly certain, again, as I mentioned at the very beginning, that when we find resistance, that it probably will not be in a selection that has all the traits that we look for in normal chipping varieties. So we're assuming that we will need to do some breeding, and we're also assuming that there are going to be different genetics that contributes to ZC tolerance, and that will require some breeding as well. So, and here we call that stacking genes. And so the whole idea here is we were trying to demonstrate a methodology for normal breeding for ZC resistance, and also for stacking genes for ZC resistance that would save 4 to 6 years, and we feel like we've demonstrated that can be done.